study of Agaricus bisporus mushroom. In this, we will get more information on network pharmacology. I welcome our today's guest speaker, Miss Nupur Gargate, who is the Siahgad alumni student and now she has taken the admission for MS in King's College of London, UK. Uh, I also welcome all the faculty members and students who have joined the session. Now I request to Pranali ma'am to introduce the speaker. Over to you, ma'am.
Thank you, Saili. Good morning to all. I Pranali Bangaya, Assistant Professor of Siahgad College of Pharmacy, Vadgaon, Pune. Welcome you all for this event. I am profoundly delighted to take an opportunity to introduce our chief guest, Ms. Nupur Gargate. Recently, she has completed her B Pharmacy from Siahgad College of Pharmacy, Vadgaon, HSC from Dr. Kalmadi Shamrao Junior College, Pune, and SSC from English Medium School, Pune. During her UG program, she has worked on projects entitled A Novel Formulation to Combat Postmenopause Symptoms in Women and other project entitled Formulation Characterization and In Vivo Evaluation of Tabernamontana Divertica Hydrogel for Wound Healing, which is still going on. She holds first prize in national level creative group presentation competition and patient counseling competition. With this brief introduction, now I would like to request our guest speaker to start today's session. Over to you, Nupur.
Hi, thank you everyone for the sweet introduction. I will begin with my presentation just a minute. Is it visible? Is the screen visible?
Yes, it is.
Yes.
Yeah, okay, so I'll start. So, good morning everyone. My name is Nupur Gargate, and the topic of my presentation today is in silico and in vitro study of Agaricus bisporus mushroom. Introduction. So, let us first begin with network pharmacology. Network pharmacology is a scientific discipline that attempts to understand drug actions and interactions with multiple targets. It analyzes the link among drugs, targets, and diseases in those networks based on artificial intelligence and big data. These are some of the advantages of network pharmacology. It predicts drug targets, establishes network of genes, and it can be used for repurposing of existing drugs as well as for designing the next generation of drugs. So why network pharmacology can be used for designing next generation of drugs is because through network pharmacology, we can understand what biological targets are present in the body and in the disease. And once we know these biological targets, we can then design specific drugs which will act on these targets. So that is why network pharmacology can be very useful in drug discovery as well. Agaricus bisporus is also known as white button mushroom. It is a commonly available species abundant in nutrients, and that is why it is consumed worldwide. These are some of the chemical constituents of it. It includes amino acids like alanine, aspartic acid, indole compounds like melatonin, tryptophan, and phenolic compounds like gallic acid, ferulic acid. Apart from these, there are several other nutrients which are present in Agaricus bisporus as well. So mushroom cell wall contains high levels of ergosterol. When this ergosterol is exposed to UV radiation, it results in the formation of pre-vitamin D2, which upon isomerization forms ergocalciferol, which is also called as vitamin D2. So basically, when the cell wall of mushroom is exposed for around 15 to 20 minutes, high amount of vitamin D2 is generated in it, more than 10 microgram per 100 gram, and thus mushrooms can act as a nutritive source. So when we expose the mushroom cell wall, because of the UV radiation of the sun, the ergosterol, which is a sterol present in mushroom, gets converted to ergocalciferol, that is also called vitamin D2. The image on left hand side shows how white button mushrooms look like, and the right hand side image shows what all nutrients are present in it. So breast cancer is one of the most common cancers, and it is the primary cause of mortality due to cancer in females worldwide. Pathogenesis is that it begins with estrogens, which form endogenous carcinogens. These carcinogens get converted to quinones, which react with DNA to form estrogen DNA adducts. And these estrogen DNA adducts cause cell transformation, which ultimately leads to breast cancer. Research has shown that there is a link between breast cancer and vitamin D because deficiency of vitamin D is linked with the onset of breast cancer. Optimal level of vitamin D in the body performs these following four functions. It reduces cell proliferation, increases cell maturation and apoptosis, suppresses inflammation, and reduces the accumulation of inflammatory cells. So, to prevent breast cancer, vitamin D should be present in optimal levels in the body. Otherwise, research has shown that it can be linked with onset of breast cancer. MTT assay is an assay which is used, it is a colorimetric assay which is performed on cancer cell lines. Main principle is that it involves the conversion of yellow MTT to purple formamazan due to mitochondrial dehydrogenase enzyme. Now, only viable cells, that is living cells, can undergo this transformation. The cells which have died due to toxicity of the sample applied do not show this transformation, and thus it indicates that the substance which is applied to the cell line is toxic. So MTT assay can predict the toxicity of compounds. So basically, in MTT assay, if the transformation happens, we understand that the cells are alive. But if this transformation does not happen, we can conclude that the cells have died due to toxicity of the applied compound. And this is why MTT assay can be used to check the cytotoxicity of certain compounds. Toxicity testing is also carried out in animals to determine the hazards of a test substance. One of the most commonly used model is the chicken eye model, and guidelines for this model are given in OECD guidance document. For the chicken eye assay, chicken eyes are collected and they are placed in saline solution. Test solution is applied to eyes, these chicken eyes, and the damage or toxicity which has happened to the eye is recorded on an hourly basis. So in my project, the aim was to study Agaricus bisporus or white button mushroom in silico and in vitro. So in the in silico part of the study, the breast cancer genes were identified and analyzed, gene network was visualized, and molecular docking was performed. While in the in vitro part of the study, toxicity of the mushroom extract was examined in MCF7 cells, and it was tested on chicken eye as well. So basically, the research was performed in two parts, in silico and in vitro. And in today's presentation, I will mainly focus on in silico or network pharmacology part of the study. Material and method, it began with text mining, which was done manually to discover the genes involved in breast cancer. Homo sapien genes were shortlisted and they were used to carry out the further steps of network pharmacology. So text mining is mainly done to understand what genes and proteins play a role in any particular disease. So since my disease was breast cancer, I used text mining to discover the genes which were involved in breast cancer. So when I went through all these genes, I selected all these genes and I used them for the next step, which was biological process and pathway analysis. Now here, analysis of the genes was done using gene codis. So gene codis is a web-based application which is used for finding annotations. Annotation is the process of describing structure and function of genes to understand their biological significance and biological process in which they participate. So mainly to get an idea of what the genes do in the body and what significant role they play, we are using Gene Codis. Gene Codis is a web-based application which we easily get free of cost on Google. And here the genes are analyzed using Go biological process categories, and then they are analyzed using KEGG. So mainly all the genes which are obtained from text mining, I first analyzed them using Go biological process, and then I obtained several genes using this, and all these genes again were used for analysis using KEGG. So here Go stands for gene ontology, and this step, Go biological process step, gives us a link between gene and biological function which that gene performs, while KEGG stands for Kyoto Encyclopedia for Genes and Genomes, which is a database that groups the genes participating in the same biological process. So Go gives us the link between gene and biological function, and KEGG is a database which groups all the genes having the same biological function. That is the difference between these two, and both are used mainly for analyzing the genes. Next step was protein interaction network, which was the major step performed. String database was used to obtain the gene network, and to analyze the network, cytoscape was used. So string mainly gives us visualization of the network, protein network, but when we have to analyze the network, we have to use cytoscape. Cytoscape is a software which we have to download in order to analyze the genes. Here string stands for search tool for retrieval of interacting genes and proteins. It is again a database which is available online, It contains information from a lot of sources, including experimental data, public data, data which has been published in research papers, etc. And it is a wide database to obtain a protein interaction network. After that, as I said, cytoscape was used. In cytoscape, to analyze the network which was obtained, we used two parameters, that were degree and betweenness, to select the key genes. Now what happens is that in string and cytoscape, we actually obtain a number of genes, but to select only the key genes or the main genes, we have selected two parameters, that is degree and betweenness. So degree mainly shows us the number of nodes which are connected to one individual node, while betweenness parameter shows the number of paths that go through one node. I will explain this a little bit more in detail when I show the actual demo of network pharmacology. But for now, we can remember that two parameters are used, that is degree and betweenness, to select the key genes. All this, whatever I explained till now, was the main part of network pharmacology. After that, to analyze the genes, we performed a few other steps, which were gene data analysis. Uh, for gene data analysis, cBioportal was used. It is an analysis tool for exploring cancer genomic data. It is a web application which is used to identify genetic mutations and alteration in each gene. Here we entered all the genes which we obtained after degree and betweenness parameters, and we entered all of these genes and we viewed the results. So mainly cBio portal is used for cancer related data. When we enter the list of genes, we get more information about what changes have actually happened in the gene, because of which that gene is playing a role in that particular disease. Another step used was drug gene interactions. This was done using the drug gene interaction database to find out what drugs are associated with each gene. Now, when we entered the genes obtained in the drug gene interaction database, we obtained several drugs associated with each gene. Now, all these drugs basically indicate the potential treatment for each gene which is involved in breast cancer. So these drugs are marketed drugs which are already available, and they act as a treatment for each of the genes which are involved in breast cancer. Then Agaricus bisporus chemical constituents were determined. Agaricus bisporus or white button mushroom was chosen mainly because it is easily available in the market. Chemical constituents present in this particular mushroom were identified and collected by referring to previous literature. So I went through all the research papers which had collected the chemical constituents of white button mushroom, and all the constituents which were obtained were the lead molecules, and they acted as ligand during molecular docking. So after we had obtained the chemical constituents, we performed docking mainly in two parts. Firstly, molecular docking of the genes was done with the drugs, and then molecular docking of the genes was done with chemical constituents. So when docking of the genes was done with chemical constituent, the genes acted as the macromolecule, and the chemical constituents, they were the ligands. Both docking of both types were done using Drug Discovery Studio 2021 and Pyrex, and binding affinity were obtained. Coming to plant material part, white button mushrooms were purchased from the locality. They were sun dried until they were completely dry, and the dried mushrooms were ground to obtain a powder which was stored properly. Extraction process was done to prepare the mushroom extract. For this, 12.5 gram of mushroom powder was taken and mixed with 20 ml of dimethyl sulfoxide. A 1:1 volume by volume mixture of ethanol and water was taken, and 90 ml of hexane was added to it. It was kept into a rotary shaker for 8 hours, filtered, and then rotary evaporator was used so as to evaporate the solvent. The dry powder which we obtained was redissolved in 15 ml of methanol, and this mushroom extract which we obtained was used further to prepare the dilutions. Now dilutions were mainly prepared in three concentration, that is 10, 20 and 40 microgram per ml, and they were used to perform the in vitro part of the study. In vitro part, MTT assay and chicken eye assay were performed. The MTT assay was performed on MCF7 cell line so as to find the cytotoxicity of the mushroom extract. And from MTT assay, we came to know about the cytotoxicity via the graph of cell viability versus concentration. And in the chicken eye assay, the chicken heads were cut and eyes were extracted. Two eyes were used for each, that is for standard 10, 20 and 40 microgram per ml. Two eyes standard solution, that is saline water solution, was applied, and on the remaining eyes the respective concentrations were applied. So for observations, we took photograph every hour. After every hour, we once the photographs were taken, At last, we applied the 2% fluorescence dye to all the eyes, and three parameters were mainly utilized, that is damage, redness, and opacity, to check the toxicity of the extract. So in this assay, mainly we took chicken heads and applied the solutions and took photograph every to check what all damage had happened in the eye. And all the results which were obtained were compared with the standard. The statistical analysis was done using GraphPad Prism, and the p-value were calculated by using Tukey's multiple comparison test. So these were the results obtained. From text mining, 44 genes involved in breast cancer were identified. All of these genes which were obtained were used in the next step. Uh, from the Go biological process, 240 genes were obtained along with their description. And all of these again 240 genes were used in the next step, for which was KEGG analysis, from which 218 genes were obtained. And now all of these 218 genes were used to create a protein network in string. So for protein interaction network, it was obtained using string database and was analyzed and visualized using cytoscape. This is how the networks look like. The left hand side image shows how the protein interaction networks looks like in string, and the right hand side shows how it looks like in cytoscape. So we can see here that there are nodes present, and these are the genes, and we can see what all relation is present between each of these genes. From Cytoscape, to analyze the network, we use Sentiscape, which is an app. It is already present in Cytoscape. And in Sentiscape, as I said earlier, degree and betweenness parameters were utilized. So these were the results of degree and betweenness which were seen. The minimum degree which was obtained was 1, and minimum betweenness was 0. Average degree and betweenness values were 18.0 and 33.93 respectively. And the maximum degree was 8.6, while the maximum betweenness was 166.6. Now, when we had obtained the network in Cytoscape, it gave all the results, it gave the degree and betweenness value for all of those genes. But in order to select the key genes, we selected genes which had value equal to or more than the average value. So whichever genes had average degree equal to or more than 18, and whichever genes had average betweenness equal to or more than 33.93 were selected. And in this process, we ended up selecting 9 genes which fulfilled this criteria. And these 9 genes were CYP19A1, EGF, PTEN, GSTP1, BRCA1, STK11, BRCA2, AR, and ERBB2. So these were the 9 genes which had average degree and average betweenness equal to or more than the average value. Now, all of these genes acted as the macromolecule during molecular docking. From gene data analysis, which was done using cBioportal, these results were obtained. It was seen that highest alteration frequency was in ERBB2 gene of 14%, followed by the PTEN gene 9%. BRCA2 gene came next with frequency 4%, while BRCA1 and STK11 had 1.9 and 1.5% frequency respectively. So this is the image. I've inserted the image here of what the data actually looks like in cBioportal. We can see that ERBB2 gene here, which is present at the bottom. There is this red colored line or red colored strip which is present. This indicates that excessive amplification has happened in the gene, because of which it is playing a role in breast cancer. Say again, for PTEN gene, we can see that there is this blue colored strip over here. This indicates that deep deletion has happened in the gene, and because of that, the gene is playing a role in breast cancer. So when we entered all these 9 genes, we got these results, which basically show what all changes have happened in those genes. Drug gene interactions, for this drug gene interaction database was used, and these were the drugs which were obtained. So when we entered the genes, actually we obtained several drugs for each gene, but for representation purpose here, I have only shown that drug which had the highest interaction score with each of those genes. So for CYP19A1, drug which had the highest interaction score was testolactone, and so on. So that is what we can see here. These drugs are actually marketed drugs which are available for treatment of breast cancer, and they are associated with that particular gene. Now, Agaricus bisporus chemical constituents, as I said earlier, by referring to published literature, 29 constituents of Agaricus bisporus were identified. They are presented in this table. These are all the 29 genes all the 29 chemical constituents of Agaricus bisporus, 5-hydroxy L-tryptophan, agaritine, alanine, and so on. All of these acted as the macromolecules for molecular docking, and the genes which had the 9 genes which we obtained, they were the macromolecules. So molecular docking, as I said, was done in two parts. First, docking was done of the genes with the drugs. So when the gene was taken as macromolecule and drug was taken as ligand, these were the values of binding affinity which were obtained. For CYP1 with testosterone, binding affinity was 8.4. For EGF with nimotuzumab, it was 7.6, and so on. And then when molecular docking was done with chemical constituents, these were the results obtained. So out of all the 29 constituents of Agaricus bisporus, we found out that ergocalciferol or vitamin D2 shows the highest binding affinity with majority of the gene. And the results are presented in these table. We can see that all these constituents have pretty less value of binding affinity. But when we look at ergocalciferol here, I have highlighted ergocalciferol here in bold. We can see that the values are very high in almost all of these genes. So from this, basically, we can draw the conclusion that ergocalciferol or vitamin D2 has very good binding affinity with all of these 9 genes. And because it has a very good binding affinity, it can actually act as a nutritive source of vitamin D. And since we also know from studies that vitamin D deficiency is linked with breast cancer onset, we can say that mushroom, because they have ergocalciferol in them, and because ergocalciferol has a good binding affinity with all of these 9 genes, it can be used as a nutritive source. These are again continuation of the same table. Again, there is no other chemical constituent which shows that high level of binding affinity with all the constituents like ergocalciferol did. Then plant material was kept under sunlight for 7 days. Extraction process was done, and weight of the dry extract was found to be 1.25 gram. Dilutions of 10, 20 and 40 microgram per ml concentration were prepared and were used for MTT assay and chicken eye assay. So these are the results which were obtained from MTT assay. The assay was performed on MCF7 cells, and these were the results which were obtained. Here is the graph presented between cell viability and concentration. We can see that for the standard solution, cell viability is 100%, which means that when the standard solution was applied, no cells died, all the cells were alive. But when you look at the 20, 40 and 80 microgram per ml, we can see that the percent cell viability is drastically reduced, and this shows that the mushroom extract which was applied to MCF7 cells was toxic, because of which the cells did not remain viable and instead they die, and because of that you can see that the graph of 20, 40 and 80 concentration are very low as compared to the standard. Then toxicity was again done in chicken eye as well, and these were the observations obtained. We took images after every hour. So for standard, when we look at standard, you can see that from 0 hour to 2 hours, no changes have happened in the eye, which mainly indicates that no changes happened, and the chicken eye is as it is. When we also look at 10 microgram per ml, there also we can see that over a period of time, no changes have happened in the eye, which again shows that 10 microgram per ml is not toxic and does not cause any toxicity to the chicken eye. But when we look at 20 microgram per ml, it was observed that after 1 or 2 hours, opacity in the eye had drastically increased, and also some amount of redness was also seen in the eye. So when we see here at 0 hour, we can see that there is no redness surrounding the eye, but here at 2 hours, we can see that there is drastic redness present in the eye. So this is also one characteristic sign of damage. And in the 40 microgram per ml as well, we can see here in this particular image at 2 hours, there is this white film which is kind of formed on the eye, and this is a characteristic sign of damage, which shows that the 40 microgram per ml concentration is toxic to the eye. Now after 2 hours, what we did was we applied 2% fluorescence dye to the eyes, to all the eyes, and when we applied the dye, we applied 2 drops. Immediately we poured saline solution over it. So after saline solution was poured, it was seen that for standard and for 10 microgram per ml, the dye was washed away. It did not adhere to the eye. When we poured the saline solution, dye got removed. But when we put the fluorescence dye and applied the saline solution over it, the dye did not get washed off like it did here. Instead, in 20 and 40 microgram per ml, that dye remained intact in the eye. it did not get washed away, which indicates that at 20 and 40 microgram per ml concentration of mushroom extract, chicken eye has shown some damage, which indicates that the mushroom extract is toxic to the chicken eyes. That is what we can mainly conclude from this chicken eye assay. Statistical analysis was done for chicken eye. Left the bar on the y-axis shows no damage, which is why at standard we can see that the bar is at 100. For 10 microgram per ml also there was no significant damage seen as such. That is why it is also close to 100. But when you look at 20 and 40, damage was observed, and it was significantly observed in 40 microgram per ml. So that's why the bar here is very less. Here it shows no damage, the y-axis, while the x-axis shows concentration. This was the damage parameter, and for redness parameter and for opacity parameter, these were the results which were obtained. So for toxicity testing, mainly, as I said earlier, 3 parameters were utilized: damage, redness, and opacity. Again, no redness, that's why at standard it shows 100. 20 microgram per ml showed redness, but 40 microgram per ml showed highest amount of redness surrounding the chicken eye. That's why here the bar is low. For opacity also we can see the similar results. Standard and 10 microgram per ml have no effect on opacity of the eye. Coming to discussion part, from vitamin D, we can say that vitamin D plays a crucial role in cell maturation, apoptosis, and in suppressing inflammation. It is discovered that around 88.7% of the breast cancer patients are vitamin D deficient, and that vitamin D deficiency is associated with risk of breast cancer. When mushrooms are exposed to sunlight, they produce abundant vitamin D. Molecular docking revealed that ergocalciferol, or vitamin D2, had the highest binding affinity with all the genes, and the obtained mushroom extract also contained some vitamin D. MTT assay, again, in MTT assay, the extract showed significant cytotoxicity on all the MCF7 cells at all concentration. Chicken eye, 3 parameters were used: damage, redness, opacity, and the scoring was done from 100 to 0. So here we can see in the table that for 10 microgram per ml, no any signs of damage, redness, or opacity. 20 microgram per ml, there was redness, damage, opacity, and for 40 microgram per ml, there was highest damage and redness. While at 20 microgram per ml, opacity was seen to be very high. Conclusion: there are mainly 5 conclusions which we can draw from the present study. First is that network pharmacology is a great way to identify the target genes which are involved in diseases. So we saw here that by using network pharmacology, we could obtain the key genes which play a role in breast cancer. So that is why network pharmacology can identify genes involved in disease. Second conclusion is that highest binding affinity was seen of ergocalciferol with all of the genes. Third conclusion, in MTT assay, extract showed cytotoxicity at all the concentrations. Fourth is that at 20 and 40 microgram per ml, toxicity was seen in the chicken eye. And the final and major conclusion which we can draw from this study is that Agaricus bisporus can be used as a nutritive source of vitamin D in cancer. So because vitamin D deficiency is linked with breast cancer, because Agaricus bisporus upon exposure to sunlight causes formation of high levels of ergocalciferol, and because this ergocalciferol has a very high binding affinity with all the genes of breast cancer, we can say that Agaricus bisporus can be used as a nutritive source of vitamin D in cancer. Future prospects. So in the near future, we plan to demonstrate the antimitotic activity of the mushroom extract, and we will do this mainly using 2 models, earthworm model and allium cepa model. In the earthworm model, the mushroom extract in all 3 concentrations will be applied to amputation region to check the regeneration of posterior parts of earthworm. So we cut the earthworm from the anterior part, and we in the clitellum region we inject all the extract, and we check whether regeneration has happened or not. So if regeneration does not happen when the mushroom extract is applied, it indicates that the mushroom extract has antimitotic activity, and it can be used as a potential anti-cancer agent. And in allium cepa model, onion or allium cepa root tip is used to check antimitotic activity. Now when these onions are grown in all the 3 concentration of mushroom extract, and when the root tips are taken and examined under the microscope, if no cell division happens in those root tips, we can say that mushroom has antimitotic activity. And because it has antimitotic activity, because it does not allow the cells to divide, it can be used as a potential anti-cancer agent. So that is why we are planning to perform these two models in the near future. So that brings me to the end of my presentation. Thank you everyone for listening. I would like to demonstrate the actual steps of network pharmacology. So here, the first step, as I said earlier, was text mining. So from text mining, I went through all the published literature, and from that literature, whatever genes I obtained, I noted them down here in the word file like this, one below the other. So these 44 genes were all the genes which I obtained from text mining. Now to analyze these genes using gene codis using KEGG, what we will do is we'll copy all these genes and we will paste them here in gene codis. So when we type gene codis in Google, this is the first link which pops up. This we will open. Organism, we will select Homo sapiens. It is selected by default. Since we want to get more information on proteins, we will select the input type as genes and proteins. And here we will paste the genes which we obtained from text mining. Now, one thing to remember here is that when we paste the genes here, we should be careful to paste the genes one below the other. There should be no comma, full stop, or space between any of these genes, otherwise we will not get the results. So we can either copy and paste all the genes here, or we can click here on browse and we can put the word file here as well. So we can do either of these. Now when we paste all the genes which we obtained from text mining here, we will select Go Biological Process and we will click on Launch Analysis. When we do this, within minutes, we will get more information on the genes which are analyzed using Go Biological Process. So just a minute. So yeah, this is what it looks like. We can see here that on the right hand side we have the genes here, and on the left hand side we can see the description which is associated with each gene. So as I said earlier, from Go Biological Process we mainly understand what genes play what role in the body. So this is how it looks like. When we click here, we obtain all the genes one below the other here, and we have to copy and paste these genes so that we can analyze them using KEGG again. So for example, in xenobiotic metabolic process, these are the genes which play a role. In heterocyclic metabolic process, these are the genes which play a role, and so on. Now we obtained several descriptions and several genes associated with each description. So what I did was I copied all these genes and I pasted them in a Word file, and I will show that as well. And this is what actually the networks looks like here in when we have analyzed the genes using Go Biological Process. So here these blue dots mainly indicate the term or the process. When we click on that blue dot, suppose we can see the process that is here, lipid metabolic process, and these orange dots here, they indicate what genes are involved in that particular process, like that. Okay, when we click here, um, we understand the biological function, that is response to organic substance, and these orange color dots, that is TFR, CYP1B1, all of these are genes which are associated with that particular biological function. This is what it looks like. Now to analyze the genes using KEGG, we have to copy and paste all these genes. So I've already done that. These are what the genes look like. I've copied all these genes which were present, and from it I obtained almost 240 genes. Now to analyze it using KEGG, again we will select all these genes, we will copy and paste them again in gene codes. To analyze it using gene codes again. Now instead of go biological process, here we will select KEGG pathways, and we will again say launch analysis. And again this is what it looks like. So for example, in the metabolism of xenobiotics by cytochrome P450, these are all the genes which play a role. And again, for all of these description, these are the genes present. This is what the networks looks like when analyzed using KEGG. Again, blue dots indicate the process, and orange color dots, they show what genes are involved with that particular process. Again, I have copied and pasted all these genes because we want to obtain a network of genes in string. So here I have copied all the genes. Again, I will select all these genes, and now we will obtain the network using string. So this is what string looks like when we type string database. This is the first thing which pops up. We'll click here. We have to select multiple proteins. So now since I have a list of proteins, I will click on multiple proteins. Again, here also I will paste the list. Here also we have to remember to copy and paste the genes one below the other, no comma, no full stop, otherwise it won't show the results. We will select organism. It is already selected here, Homo sapiens. And we will click on search. Now it gives information about all the genes which we have entered. We will click on continue, and we will obtain the network. So this is what the network which I showed in the slide actually looks like. These are called nodes. And from here, in order to, in string actually we cannot analyze this network as such, but when we have to analyze the network, we have to transport this network to Cytoscape. So for that purpose, we have to download Cytoscape software. We can see here that there are link present between these nodes, and they all have some different colors. That also has a significance. So for example, when there is a blue color present between two nodes, it shows that that interaction is derived from curated database. When we see a pink color present between two nodes, we can see that the genes are linked to each other because they're experimentally determined, and so on. This is what networks looks like. When we click here on export, we will click on send network to Cytoscape, and it will immediately send the network to Cytoscape. So I actually don't have the recent version of Cytoscape with me, so it is not actually transferring that to Cytoscape. But when you click here, it will immediately send the network to Cytoscape, and this is what network looks like in Cytoscape. This is how it looks like. When we as I said earlier, we had utilized two parameters, degree and betweenness. We click here on apps. There is Centiscape here, and there are several options present here: diameter, degree, radiality. Out of those, I had selected degree and betweenness based on the literature search which I had performed. I clicked on undirected network, and I said start. So to save time, I've actually done this step already, but all you have to do is click on degree and betweenness, and you have to just click on start, and within 2 or 3 minutes, it will show the results. So this is what the results look like here. We can see when I just place my cursor here, we obtain the minimum, maximum, and average value of betweenness. So as I had shown in that table, that the minimum value of betweenness is 0, maximum value is 166, average is 33.93 for betweenness. Similarly, for degree, when I place my cursor here, it tells us again minimum, maximum, and average values. So here at the bottom, we can see that there are genes, name of these genes, and these are the degree and betweenness values for all these genes which are shown here in the network. Whichever genes had value equal to or more than the average value of degree and betweenness, I selected those. So out of all of these, whichever had value equal to or more than average was selected, and that is how I ended up selecting 9 genes. So this was the main part of network pharmacology. And when I obtained those 9 genes, I then analyzed them using CBio portal, and also using drug gene interaction database. So this is what CBio portal looks like. It is mainly used for cancer related data. We will in this there are types of cancer according to the organ. So I will click here on breast. Here I will click on MSK nature cancer cell. So I have selected this particular uh, type out of all the available ones because I had referred to some literature and in that they had used this. So I'll be doing the same, and then we will click on query by gene. Here we have to enter all these genes, all the 9 genes which we had obtained. These are those 9 genes. And we will paste them here, one below the other again. And we will say submit query. And this is what it looks like. There are several tools available here from which we can actually understand the changes in the data. And like that, we can come to know about the changes which have happened in that data. Again, now for analyzing the genes, we also use drug gene interaction database. Here in drug gene interaction database, again we have to enter the genes. We will click on search drug gene interactions. Again, here also I will paste those 9 genes which are obtained and click on find drug gene interactions. So here, as I said, for each drug there are for each gene there are several drugs which we obtained, but I had represented only those drugs which had highest interaction score. And this is what it looks like. So for example, CYP19A1, for this gene, these are the drugs, standard marketed drugs which are available, which target that particular gene. Similarly for EGF, for PTN, and for all these 9 genes, we can see here that there are several drugs which are present. Now, CBio portal and drug gene interaction database, both these steps were performed so as to gain more insights into genes and into what drugs are present in the market. This is not really a part of network pharmacology, but I have included it in the study so that we could get a bit more idea about what drugs are present and what all mutations have happened in the genes. So that was all about network pharmacology, and I will end my presentation here. Thank you, everyone.
If anyone has any question, please feel free to ask.
Hello, Nupur.
Yes, ma'am.
You mentioned something protein networking, and before that, one slide, one concept was
Yeah.
there. I'm not getting that term. Will you
Uh,
please explain that particular term?
just a second, ma'am. I'll share the PPT again.
Yes, yes, a slide before protein networking.
Okay, just a second. Ma'am, was it this slide?
Just one slide. No,
No, or was it this?
gene data analysis. Before that, before this
Yeah.
one.
Yeah, this.
Okay, done.
Again, go back. Little
Yeah.
bit back. Before this one. Before this one, result was there. Okay.
No, yeah, till here it was material and method, and from this slide I've started the results.
Okay, go next. I don't know. I'm not getting that term. I forgot to just note it down. It was totally new and
Okay.
yes,
So was it here somewhere?
thank okay, let it be. I'm not getting that particular point. Just
Oh,
so
okay.
I once if I will come across that particular point, then I obviously I will get back to you.
Sure, ma'am. Yeah, no problem.
Okay. And the softwares which you have mentioned, these are free to use or we need to pay something?
No, ma'am, all of these are free, and all of these are actually web applications as well. But only Cytoscape you'll have to download, but that is also again free of cost.
Okay. So, how is there any method to correlate the dose which you have used on the chicken eye? Can you extrapolate to the human activity or the other animal activity?
Yes, ma'am. So just a second. So what we have done with chicken eye is that first of all, to test the toxicity, chicken eye is a very common model, and it is very easy as well, and it is inexpensive. So what we have done is that mainly to see if there is toxicity topically when applied to the chicken eye. We have used the chicken eye model. So that is why when we apply it topically, like we just poured 1 or 2 drops of the respective concentration, we saw that damage had occurred. So this is mainly to test the toxicity of the particular compound.
Okay, just to check the toxicity, nothing else.
Yeah, only to
And
yeah, determine toxicity. Yeah.
you have used 10 microgram per ml, 20 microgram
Yeah,
per ml
and
solution
40.
like that,
Yes,
and
but at
40. So
10,
out of that,
yeah,
okay, so just to check the whether it is toxic or not, and at
no,
which particular stage it is okay.
yeah.
Okay, okay.
So we
Thank you so
actually
much.
at 10 also, yeah, we at 10 microgram we saw no damage as such. So we only saw damage at 20 and 40, indicating that these two concentrations are toxic like that.
Okay, okay, okay. Okay, thank you. Thank you.
Yeah, thank you, ma'am.
Hello, Nupur.
Yes,
Hello. I have one question. Have you done the
yes,
authentication
ma'am.
of mushrooms?
Ma'am, actually, we did not authenticate it.
But you should now.
Yeah,
And
yes, ma'am. That's
and
it. We yeah, kind of realized a little later.
okay. And the second point is, can you go to your extraction process, the previous one?
Yeah, just
Initially,
a
you have
second.
mentioned the solvent system for the extraction.
Yes. Yeah, ma'am. Here it is.
So I'm unable to understand why you have chosen the two different phases, these solvent systems, because the one you have chosen is a polar system, and the second
Yeah,
one it's a mixture of polar and non-polar. So
non-polar.
what
Yes.
is the particular reason behind this selecting this type of
But
the phase?
yeah, ma'am. So actually, so before the extraction process, we had done molecular docking, right? And at that time, I came to know that vitamin D or ergocalciferol had the highest binding affinity. So actually our goal was to extract vitamin D in the extract. So for that, I looked up the extraction process which had extracted vitamin D in mushroom. So for that, when I referred to research paper, this was a particular process which I found, and they had done exactly the same steps, only they had differed the quantities a little bit. So that is why I have exactly followed that particular research paper. So their aim was actually to extract vitamin D from mushroom. So that is why we followed this process.
But you can take only hexane, no? So what is the purpose of taking this aqueous phase for extraction? Because it will not isolate vitamin D, right? Because since it is
Yeah.
a non-polar, soluble in the non-polar solvent, so I think there is no use to take the aqueous
Okay.
phases which you have mentioned over here.
Yes, ma'am. Okay. So actually, we just
So
follow.
just
Nice presentation, Nupur. Presentation
Thank
is
you,
very
ma'am.
nice. Hmm.
Thank you so much.
just sorry, Nupur, just to add, madam,
Yes.
actually, we done network pharmacology, we done molecular docking, and based
Yes.
on the argocalciferol, we decided to go for the extraction. So we knew that we are not going
Okay.
to able to identify if there is vitamin D present in our extract or not. But we referred
Okay.
article to find out if the procedure is available where vitamin D can be extracted. So we referred the
Okay.
article which have shown that the vitamin D can be extracted with this mushroom. So we use the same procedure. We actually have not done any changes in the procedure. So we
Oh,
are assuming that that extraction is actually containing vitamin D.
okay. Okay.
Yeah.
So thank you.
Yes. Any other questions?
Any questions? Anyone? Any student, faculty? Thanks Nupur for accepting the invitation from Sendidi and
Thank you.
Swalife and giving this presentation. It was a nice presentation. At the same time, you actually done a good work in the network pharmacology, and it's not only that you have done, you actually have done a presentation as well. Definitely, we will come back if there are any more questions from my side or from other faculty and student, and hopefully we will,
Yes,
refer
ma'am.
to you, and hopefully we'll get help from you regarding network pharmacology. And
Definitely,
I
sir.
like to wish, I like to wish you best of luck for your master's degree.
Thank you so much, sir. Thank you for inviting me, actually. Thank you.
Uh, Over to you, Siley.
Now I request Sudarshan Shinde to propose the vote of thanks.
Good afternoon to all. I feel pleasure to purpose vote of thanks on behalf of our college Sinhagad Education Society, Sinhagad College of Pharmacy Pune for arranging this session. It was very beneficial and I hope it will give us immense knowledge even if in future when we remember it. Thank you to our today's speaker, Miss Nupur Gargate for sharing her valuable time by guiding and demonstrating about the network pharmacology and her work on the mushroom. I wish her for all the best for her master study. So once again, we are grateful to the enlightened by you. I would also like to thank our founder president, Professor Yemin Navle sir, founder secretary, Dr. Sunanda ma'am, vice president admin, Miss Rachna Navle Ashtekar ma'am, vice president HR, Mr. Rohit Yam Navle sir, for their motivation. I would also like to express my humble thanks for the principal Dr. Rajesh Patil sir for arranging and planning them and making this program successful. Your support and guidance lead us on the path of success. I would like to extend my gratitude to Dr. Praveen Bade sir and our teacher for their active participate in making this event successful. Once again, thanks to one of all who give their contribution along with. Thanks. Now I request everyone please turn on their cameras for the group photos.
Thank you all.